Our top story is an update from the world of evolution. If you know anything about evolution, you know it's a fairly slow process when measured from a human perspective. However, bacteria generally reproduce very quickly, which unfortunately has led to the very serious issue of antibiotic resistance. This fast reproduction and adaptation can also be helpful to scientists that study evolution, which brings us to the 25-year-old experiment happening at Michigan State University. The setup is pretty simple. 12 flasks containing E. coli, low concentrations of glucose to feed them, and lots of citrate. Now the citrate is the important part. The inability of E. coli to metabolize it in the presence of oxygen is a defining characteristic, separating it from other bacteria. It's estimated E. coli's ancestors lost that ability about 13 million years ago, but after 33,000 generations, one of the flasks became cloudy. That was the beginning of E. coli's evolution. The MSU experiment is now over 56,000 generations in, and the bacteria are now reliably feeding on the citrate. Scientists continuously kept track of each population's genetics, so we have a play-by-play of how evolution happened in this experiment. First, 2 mutations occurred as early as 20,000 generations in, which likely set the stage for this adaptation. Next, a dormant gene was duplicated, and the copy was activated in this bacterial genome. This gene coded for a citrate pump protein. Finally, several other mutations occurred, allowing citrate to be fully metabolized, and this included further duplication of the pump protein gene. The ability to track this process so precisely gives scientists a tremendous insight into the mechanisms of evolution and demonstrates the power that small, successive changes have over many generations. Our top story comes from the field of material science. Researchers at the University of Pennsylvania have developed a new method for growing organs. Remember last week when we discussed how regenerative medicine had the potential to regrow entire organs, but that there were many technical challenges? Well, a big challenge is vasculature. That's mimicking the network of blood vessels that would be in an organ. Current methods involve essentially printing the organ layer by layer, including hollow vessels. The problem is printing these structures creates seams that can't easily withstand the pressures of the nutrient formula being pumped in. Another issue is that certain cell types, such as liver, can't usually survive the normal layering method. To overcome this issue, the researchers actually printed the vasculature first, a network of hollow filaments. They were made from a mixture of glucose, sucrose, other structural sugars, and reinforced with a coating of biodegradable plastic. Testing with this method went extremely well due to a simplified procedure. Once the filaments are printed, a cell solution is just poured into a mold. Now these filaments are initially quite sturdy, but easily dissolve as the vascular channels form, infusing them with blood vessel cells and even naturally sprouting capillaries. By the way, these tests were done with cultured human cells, including liver, and are approaching clinically functional levels. Obviously, there's still much research to be done, but it's still an important step. It's especially encouraging that the researchers are embracing the open-source ideas behind the 3D printers they use. We begin with a story from the world of biotechnology. As you may know, human breast milk has some amazing properties that help develop a baby's immune system. This can be partially attributed to specialized sugar molecules that encourage the growth of healthy gut bacteria in the newborn. So a researcher at the University of Illinois wanted to study 2FL, the most abundant kind of special sugar, using a piglet model to understand the specific mechanism by which the immune system was boosted. Unfortunately, 2FL costs about $100 per milligram, so the study feeding it to piglets would cost around $1 million. However, the University of Illinois also had some awesome metabolic engineers, who then helped out by creating a strain of E. coli that produces this human sugar. This was possible because E. coli already synthesizes a precursor to lactose. They boosted that metabolic pathway and inserted a gene for lactose formation, followed by another that allowed the bacteria to turn lactose into 2FL. Using this, scientists can produce 1 gram for every liter of E. coli broth. The same amount would have cost $100,000. Now it can be produced in the lab with a relatively simple process, so it can be properly studied. But things won't stop there. If research shows the health benefits of the sugar to be significant, it could be synthesized on a larger scale and added to baby formula, maybe even added to soldiers' food to boost their digestive health, among other potential applications. Next is a story from the world of neuroscience. The Blue Brain project out of Switzerland has made significant progress in the development of neurocircuitry computer models. The Blue Brain project is attempting to do what many fields are doing, take the vast amount of very specific data and turning that into a useful computer, with the overall goal of stimulating entire brains, starting with simpler animals but eventually moving on to the human brain. A critical step toward that goal is understanding how synapses form. To review, synapses are the crystal points of near contact between neurons, allowing an electrical impulse to pass from the dendrites of a neuron to the axon of the next. A major question was, how do synapses form? Do the branches of neurons grow and connect randomly, or perhaps neighboring cells emit chemical signals to guide synapse formation? So to test this out, the BBP team reconstructed thousands of different neurons in the computer, based on over 20 years of data examining brain tissue. They then created a model with 10,000 of these virtual neurons, in random locations and orientations, to map out where the synapses end up. Comparison with real neural circuitry showed that the model was between 75 and 95% accurate, and this suggests that for the most part neurons grow physically independent of each other. However, there were some exceptions where synapse formation seemed statistically guided, but that adding those to the model's parameters could make it even more accurate. Ultimately, this is a big advancement towards stimulating neural structures and our understanding of the brain. We begin in the world of biotechnology. A strain of wheat genetically engineered with a peppermint gene is being tested in the UK. This peppermint gene isn't about giving wheat flavor, but producing a pheromone signal undetectable by humans. The notorious pest aphids, on the other hand, can detect it. It's the signal they use to warn of an attack from predators. The hope is that, if this works, wheat growers could cut down or even eliminate pesticides. Naturally, protesters against GM foods have come out against this, pointing out that the aphids may adapt quickly. While it's certainly possible the aphids could start ignoring the chemical signal, it doesn't just repel them. This pheromone also attracts parasitic wasps, which are one of the aphids' main predators. And the project is different from other similar GM foods in that the goal is repelling pests rather than killing them. For some opinion, this method certainly seems like a step in the right direction for a greater acceptance of GM crops. But again, this is a test. 8 small plots of wheat are being grown at the Rothamsted Research Agricultural Institute in Hertfordshire, where they'll be observed over the summer to see if the gene modification performs as expected.